Welcome to Making the Rounds, a podcast by the American Medical Association. I'm Maria Lisa, last year's winner of the AMA Research Challenge, which is the largest national multi-specialty research event for medical students, residents, and international medical graduates. Today's interview features one of this year's 5 finalists for the 2022 AMA Research Challenge, interviewed by AMA senior news writer Brendan Murphy.
Hello, and welcome to Making the Rounds by the American Medical Association. I'm Brendan Murphy, senior news writer at the AMA. Today, we are joined by Leela Biswas. Leela is one of the 5 finalists for the AMA Research Challenge. Hi, Leela. How are you today?
Good. I'm doing well. Thanks so much for having me.
We are very excited to have you. Leela is pursuing an MD PhD at Rutgers Robert Wood Johnson Medical School. Her submission poster for the Research Challenge is entitled Decoding Pregnancy Loss, Validating a Novel Genetic Biomarker of Poor Egg Quality. Let's get into your research, Leela. Can you tell us a little bit about this topic, why it appealed to you, and how you got involved in the AMA Research Challenge?
Sure. Happy to discuss. So my research really deals with the disease of infertility, and it's a topic that's not widely discussed. I think because the experience of miscarriage and of infertility is really painful for individuals and for couples. As some background, roughly 1 out of every 10 female identifying individuals in the United States are diagnosed with infertility. So it's a lot more prevalent than people realize, and it's incredibly devastating for individuals who want to get pregnant but can't. And about 25% of pregnancies end in miscarriage. So really strikingly common. But the leading genetic cause of miscarriage is something in the embryo called aneuploidy. And so aneuploidy is a chromosomal abnormality in the cell. Basically, like the cells have the wrong number of chromosomes, an incorrect number of chromosomes. And when you have an aneuploid egg and it gets fertilized by a sperm, it nearly always leads to miscarriage via an aneuploid embryo. But the only biomarker that we have for this is maternal age. And it's a J-shaped curve, which means that on average, people, as they get older, tend to develop more aneuploid eggs. And that sort of skyrockets around 30 to 35. And the chances of a successful pregnancy go down with that. So maternal age is actually a really effective predictor for egg aneuploidy, but it doesn't work for everyone. So there are individuals who are at an advanced maternal age and have very few aneuploid eggs, and people who are very young and have a lot of aneuploid eggs and unsuccessful pregnancies. And so if age isn't predicting aneuploidy for these individuals, there has to be something else at play, right? Some other factor. And so we hypothesize that there's like a genetic contribution as well, that some people have more egg aneuploidy than you'd expect for their age because of their genetics. And if we could identify those genetic factors, then you can imagine what we might be able to do with that. It's to identify a predictive genetic biomarker so that we could identify them not via invasive testing or just sort of by having them get pregnant and then lose the pregnancy over and over again, but by a simple blood test well in advance of actually trying to conceive a child. And so the thought is that a physician in the clinic, before somebody has decided to get pregnant, sits down with the patient, and does a blood draw, runs a genetic test for biomarkers, combines that with maternal age, and then they can together design a care plan and a pregnancy plan that works for them. So maybe that is starting a family earlier if there's somebody who is going to have a lot of aneuploid eggs at an earlier age than you'd expect, or it might be egg donation, it might be IVF, it might be adoption. It's whatever makes sense, both clinically and personally, for the patient and the physician, if that makes sense. So what we did here was to take a cohort of patients, pull out those individuals who had statistically extreme rates of egg aneuploidy relative to maternal age, and then use whole exome sequencing to identify genetic variants that were enriched in those high aneuploidy people. And then we took those variants and we asked, well, do those variants really cause aneuploidy in those patients, or is that just a coincidence that some high aneuploidy patients have these genetic variants? So we tested those variants in actual mouse eggs. So we overexpressed the variants in mouse eggs using microinjection and asked if the variants in question could disrupt meiosis, basically mess up how the egg divides and segregates its chromosomes, causing aneuploidy. And in fact, we found one that did. And then the question was, well, so that's in a dish. Those are cells in a dish, and you're overexpressing them, so they're not at physiological levels. So what happens if it's in a whole organism? What happens if it's at a physiological level? Does that variant still cause egg aneuploidy? And so we generated a novel knock-in mouse with the help of the Rutgers Genome Editing Core using CRISPR. And we took that patient variant in question and knocked it into the mouse. So the mouse has the same patient variant, and we asked, does this mouse have increased aneuploid eggs? And it turned out it did. Compared to its age-matched peers that had a wild-type copy of the gene, the mice with the mutation in question that we found in patients had an increased rate of aneuploidy. So it was really exciting, and our hope is that this lays the groundwork for then developing those biomarkers that we can use to predictively identify patients well in advance of attempting to conceive. And this appealed to me because I previously had volunteered for 2 years with children with developmental disorders, as well as did research in cancer biology. And so as I went through my education, I learned more and more about the genetic basis of developmental disorders, as well as how genetics could be applied to many fields of medicine to understand the origins of disease. And so I was obviously very excited to learn about this project where I get to learn about the unraveling of genetic data to really understand how disease, and in this case, egg aneuploidy and miscarriage, can arise during development. And I was also able to learn how to use broadly applicable scientific tools like mouse models and classical genetic approaches, high resolution confocal microscopy. You'll have to check out my poster to see those pictures, and dig deeply into cell biology and genomic stability, which are broadly applicable biological concepts. And so I anticipate that I'll be able to use those tools as a physician scientist to tackle the most pressing diseases when I'm a fellow or an attending.
Well, that certainly is research and work that could really affect the lives of many, many patients and certainly seems translatable. What were some of the challenges you encountered in doing this research, Leela?
Sure. So the biggest challenge, I think this is a really multi-pronged project with a lot of moving parts. So we have computational collaborators, as I mentioned, that we pull data from biobanked genetic sequencing data from patients. So we have computational collaborators who are able to analyze those data and extract the most enriched variants, and we collaborate with them closely. And then we're sort of the biological end. And so we have to have a lot of back and forth between the two groups. And we need to be able to closely collaborate and also be able to comment on each other's work because they understand the computation, we understand the biology and the clinical aspects of it. And so there's a real back and forth there in terms of the coordination. In addition, I'm using a mouse model and a novel mouse model. So it takes a really long time to get a new mouse model up and running. So mice have a gestation time of about 20 days, and then they take another 6 weeks to be sexually mature. So it's a long time between having the mouse that you need born and being able to actually do the experiment that you want to do. And then you need multiple replicates, and sometimes experiments don't go as planned. And so that really slows down the process and makes it very labor intensive, but extremely rewarding because the mammalian model is essential, I think, in the setting of this experiment. And then also all of the challenges of research. So often experiments don't go as planned. You have to repeat things. And so it's a lot of that troubleshooting and reworking and learning so many new techniques. I had never worked with a mouse before joining this lab. Learning high resolution confocal microscopy with $1 million microscope. That was really tough and scary. And it takes a lot of failure and being able to pick yourself back up and learn and ask questions and go to others for help and do all the research to figure out how to make things work. And so that was really, it's that difficult process of science, of failing and then just trying again in smarter, hopefully ways, to eventually succeed.
What advice would you offer to medical students who are conducting research on a project like this?
So first, right off the bat, you need to find a project that you're really excited about. If you don't have something that you're super thrilled to be doing, it's going to be really difficult to do it as you experience those failures and challenges in science. I'm thrilled to be doing my project, and I'm really hopeful about how we can positively impact the lives of patients, and that really drives me forward every day. So find something you are excited and deeply passionate about. Second, be prepared for challenges. So as I mentioned before, research has tons of failures and hiccups and troubleshooting before your success. during your becoming successful, and then after you've been successful, you will just continue to find failure. And so you have to be able to get knocked down, fail, pick yourself up, learn from it, and keep going. Be prepared for that. And then also be ready to learn all the time. So, as I mentioned, I had learned a lot of new techniques in doing this project, from the microscopy to the mouse genetics, to oocyte biology to 4D image processing software. There's so much to learn in a new and exciting project. The more exciting your research is, probably the more you'll be learning as you do it. And I think that's the key to a lot of science and probably a lot of clinical medicine, although I'm still a student. Um, the field of every field of medicine is just accelerating so rapidly. There are always new techniques. There's always more data to be found. And if you are static, you fall behind. So you just have to keep being like a sponge and learning more and more and more and accelerating. And you can't be afraid of that. You're not going to get everything, but you just have to keep learning and being excited about your science and about your medicine, and remember why you're doing it, which is for the patients.
You took care of the nation. It's time for the nation to take care of you. The AMA stood by America's physicians and patients during the pandemic, and we're not stopping there. We're fixing prior authorization, leading the charge on Medicare payment reform, supporting telehealth, fighting scope creep, and reducing physician burnout. It's time to rebuild, and the AMA is ready. To learn more about the AMA Recovery Plan for America's Physicians, go to ama-assn.org/time to rebuild.
Can you talk about how these findings could maybe contribute to a change in the way some patients go about having a child?
Sure. So what we did was identify a genetic variant that increases the rate of aneuploidy in mice and is enriched in patients with high rates of egg aneuploidy. And so our hope is that this will lay the groundwork for this variant to become potentially a genetic biomarker. And so the idea is that prospectively in the clinic, before people are ready to have a child, before they're conceiving, that they would receive a genetic test for this and other biomarkers that are well validated, well studied in large cohorts long term, which we haven't done yet. So that's an important next step. So they would receive genetic testing, and that the results of those genetic tests could be combined with maternal age data, because that's still a critical, important, fundamental factor in causing egg aneuploidy. And then the patient could talk to their physician as well as their genetic counselor about the results of those tests to determine when is a good time and what is a good strategy for them to achieve a successful pregnancy. So for example, if there's a patient who has a lot of variants that predispose them, or one variant that predisposes them to having a high rate of aneuploidy at a young age, so say, for example, maybe they'll have a lot of aneuploidy at the age of 28, which is usually an optimal time to have a pregnancy based on what we know about maternal age outside of variants. So if they have a variant that causes them to have that high aneuploidy, maybe they want to freeze their eggs at an earlier age before their aneuploidy increases. Maybe they want to consider egg donation. And all of those interventions would have to be tested empirically to determine like the risk and benefit long term in patients. Right now, we're just laying that groundwork. But the idea is that this would be integrated. So genetics would be integrated with maternal age in the clinic by a physician with the patient involved to design really a personalized care plan. So it's strategic pregnancy planning to avoid some of that heartbreak, to avoid some of the pain of miscarriage that so many people are facing.
We talked about the big picture plan, how this applies there.
Yeah.
What are the next steps in this work?
Sure. So there's like short term and there's long term. And in the short term, in terms of what I'm doing in my next steps in my thesis work, I'll be using this novel knock-in mouse line as well as a couple other mouse lines that we have in the lab to explore the mechanisms behind mutation that we identified, KIF18A MDM, how that causes aneuploidy in eggs. And that sounds like a really basic fundamental science question, which is always incredibly valuable to explore the basic side, but it's also important, I think, clinically and translationally. So when you have a mutation in a gene that causes an amino acid substitution or change in protein, learning more about that specific region of the gene and protein can help you understand how other mutations that people have can affect that same gene or protein and then long-term lead to potentially a similar phenotype. It doesn't mean that necessarily if somebody has a mutation at spot 25 that another person at spot 26 will have the same disease as person A, but it does mean that perhaps if you find another patient with the same phenotype who has a mutation nearby, that might be a good candidate to study, that there's established evidence that might lead you down that road. And so really exploring those mechanisms and understanding how the protein works and how a given mutation can disrupt the function and lead to a specific phenotype is really important, and can also lead to the development of targeted therapies down the road. And so that's what we're going to be exploring next, really digging into those mechanisms of how KIF18A MDM causes aneuploidy in mouse eggs. And then long term, the lab wants to identify more mutations that cause aneuploidy and validate them in knock-in mouse models. And that work is ongoing in multiple genes using the same pipeline. So basically identifying mutations that are enriched in high aneuploidy individuals and validating those in mouse models. And then downstream, the really critical work in translating this is going to be validating this in a large human cohort. And so our hope is to really identify those causal variants in our population and with our biobank data, biologically validate those in mice, and then clinically validate in large human cohorts and hopefully translate this work.
It certainly sounds like a process, but it does sound like you're on the right and exciting track.
Thanks.
You yourself are in the process of forging a career toward becoming a physician. How does this work impact your career trajectory, or does it?
Yeah, definitely. I mean, the big thing is the intensive research training combined with my clinical training is incredibly valuable. It has armed me and equipped me with not only the scientific thinking and the approach to rigor and reproducibility that are going to be critical as a physician scientist, but also a number of useful tools such as classical mouse genetics, such as high resolution microscopy, imaging, being able to really see inside the cell. We can do that. It's amazing. So I'm going to be able to use those tools long term as a physician scientist in the future. And then, more in terms of the topic of my research. So my research really focuses on precision genetics. And so really digging into large amounts of sequencing data from individuals to identify genetic variants that we can correlate with phenotypes and disease states in the hopes of identifying therapeutics, diagnostics, and prognostics. And so that's something that has really shaped where I want to go in my career. We have so much omics technology now, from the genome and exome to the proteome and beyond that, transcriptome, and on and on and on. There's so much data that we can pull from each of us, from each patient, in each of ourselves, to identify how what makes me me and what makes you also makes me have one disease and you potentially have another disease, and how that shapes how our treatments might be different, and what might be optimal for me might be different than what's optimal for you to achieve a healthy and happy life. And so really driving towards precision medicine, that has really been, this project has really shaped my desire to become somebody who is a physician scientist that specializes in precision medicine. That's where all the fields of medicine are going, I think, in some way, as we gain more and more data. And I'm excited to be on the forefront of that and be part of that army of precision medicine physicians really optimizing care plans for patients.
That certainly is the future. What else should our listeners know about your journey in medicine?
Sure. My journey has been like the weaving together of many valuable experiences. So I spent 2 years volunteering with children with developmental disabilities in high school. And then I did intensive research in undergrad at Thomas Jefferson University's Kimmel Cancer Center in college. And then my medical school studies have really integrated for me all of these myriad experiences in that biological and clinical context to help me identify my passions. So along the way, I've also had really great mentorship and collaborations. And so together, all of this has led to exciting MD-PhD research, and for which I'm fortunate to have received NIH funding in the form of an F30 predoctoral fellowship. I'm extremely lucky to be doing the research I'm doing at Rutgers, and also to have received funding to continue my project.
So as you know, and I think our listeners will know if they've listened to other episodes of this podcast, The Research Challenge winner is awarded $10,000 from our sponsor, Laurel Road. What would you do if you won that money?
Yeah. Well, there are a lot of expenses involved in an 8-year graduate MD PhD program. It's twice as long as the average medical school. So I would probably invest in supporting some of those educational expenses so that I can become a physician scientist and then meaningfully give back to my patients and my community. I'm very lucky to be participating in the Research Challenge and to be advancing my academic career so that I can do meaningful work for patients.
It does seem to have a lot of translational value and really could affect the lives of patients going forward.
Thank you. I appreciate it, Brendan.
Absolutely. This is important work being done here. Thank you so much for joining us and sharing it with us.
Thank you so much for having me. I appreciate it. Have a good day.
And a quick reminder for our listeners, you can see Leela's research poster as well as those of the other finalists in the Research Challenge at that Research Challenge website, which is ama-assn.org/research22. And of course, be sure to tune into the finals of the AMA Research Challenge on December 7th to see all 5 finalists present their work to a panel of expert judges for the chance to win that grand prize. This has been Making the Rounds, a podcast by the American Medical Association. I'm AMA Senior News Writer Brendan Murphy. Thank you for listening.
Join us on December 7th at 7 p.m. Central Time to see all 5 finalists present their research to an elite panel of judges. The overall winner will receive a $10,000 grand prize, sponsored by Laurel Road. For full details, visit ama-assn.org/research22.